Welcome, and thank you for joining us for this special conversation with Cure Alzheimer's Fund leadership. I'm Danny Harper, Vice President of Development at Cure Alzheimer's Fund, and I'll be moderating today's discussion. We are so grateful for the many supporters, partners, and members of the CureAlz community who've joined us today. While each of you have your own personal reason for joining, we are all united by our shared goal of ending Alzheimer's disease. CureAlz was founded in 2004 by the Morby, McCants, and Rappaport families who were eager to accelerate Alzheimer's research with the highest probability of preventing, slowing, or reversing the disease. Today, 20 years later, a cure for Alzheimer's remains elusive, but the remarkable progress being made by CureAlz-backed researchers across the globe is cause for hope and celebration. CureAlz is known for supporting novel hypotheses and bold ideas. Our grants allow researchers to test new ideas, leading to a greater understanding of the causes of Alzheimer's. Thanks to our board of directors, trustees, and a core group of other donors who direct their donations to our operating expenses, 100% of general donations go to our research program. Driven by thousands of people who are motivated to help current and future Alzheimer's patients and families, CureAlz funds cutting-edge research that builds new bodies of knowledge and pushes the field toward a cure. Since our inception, we have funded over $260 million to more than 1,000 research grants worldwide. I'm truly excited to reflect on these achievements and to look ahead during today's conversation. Before we begin, a few housekeeping items. We will conclude today's one-hour program with a question and answer section. We welcome you to submit questions using the Q&A feature at the bottom of your Zoom screen, and we will answer as many as we are able. We will record today's presentation, which will be shared with all registrants once it is available. Now, it's my pleasure to introduce our speakers. First, Meg Smith, CEO of Cure Alzheimer's Fund. Meg joined CureAlz over 10 years ago to lead our research management team, and in 2023, she was appointed CEO. Meg leads the organization's strategic vision, partnerships, and fundraising efforts, ensuring CureAlz remains focused on bold, high-impact science aligned with our mission to end Alzheimer's disease. Welcome, Meg.
Thank you, Danny. It is great to be here today.
Thank you. Joining Meg is Dr. Natasha Hussain. Natasha joined CureAlz in 2025 as Executive Vice President of Research Management, bringing deep expertise in advancing neuroscience and cross-disciplinary research initiatives. Most recently, she served as the inaugural Associate Director of the Knight Initiative for Brain Resilience at Stanford University, where she led strategic programming and partnerships to accelerate discovery in brain health and longevity. Prior to Stanford, Natasha was Scientific Director of the Kavli Neuroscience Discovery Institute at Johns Hopkins, fostering collaboration among neuroscientists, engineers, and data scientists. Natasha earned her Bachelor in Science in Biology and Environmental Science from McGill University, followed by a Ph.D. in Neurology and Neurosurgery. Natasha then completed a postdoctoral fellowship at the MIT Picower Institute for Learning and Memory. We are incredibly fortunate to have Natasha on the CureAlz team, where she oversees our global research portfolio and works closely with scientists and advisory leaders to accelerate new discoveries, many of which she will highlight today. Welcome, Natasha.
Thank you, Danny. It's a real privilege to be part of the CureAlz mission, and I'm looking forward to today's conversation.
Thank you so much. Meg, we'll get started with you. As you reflect on 2025 and think about the year ahead, what stands out to you the most?
Wow, that is a tough one to say, to respond to quickly, because 2025 was an incredibly tumultuous year. We could talk this entire hour about all of the challenges facing American science in 2025. That's not why we're here, so that's not what I'm going to do. But it absolutely was a public context that was just a real sea change from what has happened in American science over the decades before this. We have built an incredible foundation for American science. Our infrastructure, our talent base, our biotech and VC entities that are ready to pick up the great discoveries that American science generates. All of this has been integral to our success as a nation and as an economy. And so it was really shocking for American science and scientists and those who care about what comes out of them to see all of this challenge last year. So while we watched all of the different challenges emerge, and there were different challenges from month to month, it was kind of astonishing how different the conversation had to be every single month. We realized that the way to make sure that we not only protected progress but continued to accelerate progress was to recognize that our mission didn't change. Because we are so laser focused on achieving a world without Alzheimer's disease, we are able to identify what we should do, what we should support, and what we shouldn't do on the basis of, does it serve that mission or doesn't it? And that focus allowed us to be incredibly resilient and to continue to unite our community around that mission. And I'm so deeply grateful to our donor community and to our scientific community for maintaining that focus and therefore allowing us to have our progress be resilient to all the challenges around us. So, although I could tell the story of 2025 as a year of immense challenge, and I can tell that story and I will tell that story, I also get to tell that story as a year of incredible progress and a year where we did not lose focus and therefore we did not lose progress. And I'm really proud of that and grateful for that across all aspects of our community.
Thank you, Meg. When you think about the challenges that face the research landscape, are there any aspects of that you want to highlight or think about that we're going to look at and address in the year ahead?
So one of the things I'm really proud of it as an organization is that as our community rallied to provide increased resources for us to provide to our scientists, we did that in real time. Most philanthropies operate on a model where they essentially bank at least some portion of what they raise every year, kind of as a rainy day fund. And we do make sure that we have appropriate reserves so that we can be a reliable source of funding for our researchers. But we immediately put the money that we raised in excess of the past last year to work. And we will continue to do that this year, and we will continue to have higher funding this year than we have had in past years in response to the crises around us. So identifying exactly how we can best use those resources in 2026 is an ongoing, an exciting challenge that I know Natasha and her team are thinking about a lot. We are at the same time trying to ensure that we continue that line of communication with our donor community with events like these so that we can explain exactly what we are doing with these resources to hold ourselves accountable as we always want to do and to share how these increased resources are actually translating into progress. So I'm excited to continue this line of communication with our donor community and to continue to hear from our scientific community how we best shape our support to meet their needs as their needs change given the context in which they're operating.
Thank you so much. Natasha, turning to you, 2025 was not only the year you joined us at CureAlz, but also an important year for scientific progress. Can you share one or two major breakthroughs from the year that stand out to you the most?
Yeah, thanks, Danny. There was a lot of progress, as Meg said, that was made last year. So I'm really happy to highlight some of the research that was done. CureAlz supported funded research was cited in over 300 papers last year. That's each one of those peer citations are a scientific progress that's meaningful advance towards Alzheimer's understanding. And I'll just talk about one example right now for why these publications matter. One of the projects that we supported was by the group led by Dr. Andrew Yu, who's at WashU. He published a paper in Science that addressed a very long-standing question or limitation in Alzheimer's research. Several years ago, we were fortunate as scientists in the community to be able to take skin cells. The discovery was made so that you could take skin cells or fibroblasts and reprogram them to become stem cells. And that was a great breakthrough because it allowed us to take these stem cells and modify them into neurons, which allowed us to look at alternative model organisms for studying neurodegeneration, neuro-related disease. However, there was one big limitation to this fantastic discovery, and that was that when you reprogram these cells, they become like newborn baby cells. They've lost all of their aging signatures. And so that doesn't work for modeling aging disorders or diseases such as Alzheimer's. So what Dr. Yu's great breakthrough was that he was able to define a mechanism by which you could still take these adult cells and directly transform them into neurons, where they would preserve their actual birth date and their aging signatures throughout. So that means this breakthrough allowed us to really have, for Alzheimer's, human neurons that behave like aged cells. And this was not just important for Alzheimer's. His discovery was really impactful field-wide to all neurodegeneration studies. So it was really a foundational advance that affects everything downstream from disease modeling to drug discovery. And what excites me about this is that we're not just making incremental progress. We're really changing the tools and the concepts and the cross-disciplinary thinking that's going to define our Alzheimer's focus in the coming year and years forward. And I'm really happy that CureAlz funded science is really pushing that momentum forward. That's exciting to me.
Thank you for sharing that. It is exciting work. When you think about that breadth of possible work that can get funded by CureAlz, can you speak to how the organization prioritizes its decision making and thinks about what to fund in the year ahead?
Yeah, that's a great question, because what we prioritize is really how we drive what we want to see moving forward. And I think the way we prioritize, in my mind, is that we look at where society is today, where society is moving, and what are the cutting edge discoveries that we're now capable of applying. So Meg gave you an example of us meeting the moment this past year in terms of our changing infrastructure in terms of science support. But looking further, more broadly ahead, last year was the year that marked what demographers are calling entering peak 65, which is a period of time where the largest cohort of Americans are going to be entering the age of 65 and onward, which is the highest age, or is the bracket with the highest risk for Alzheimer's disease, because Alzheimer's disease risk increases with age. So for CureAlz, in terms of defining priorities, the question really becomes, how do we meet that moment scientifically? We're really fortunate, I think, in the past 10 years to have really be at like a scientific inflection point where we've made enormous strides in big data, computational capabilities, unprecedented abilities to image cells with unprecedented precision, and also modulate cells and proteins and molecules unlike we've ever been able to do before. So some of these buzzwords that I'm sure you've heard are things like CRISPR or CAR-T, proteomics, lipidomics, spatial transcriptomics, neuro AI. All of these advances are leading us to really exciting areas in science that can be prioritized. And the ideas or areas that I'm really excited about looking forward this year are aging and brain resilience, because as for the majority of Alzheimer's research, we've really been focused on what increases risk. But when we start looking at an aging population, it is clearer and clearer that we can start asking questions about what is resilience and how is it that we can remain cognitively intact as we age. And that's an important question, because there are populations that are potentially at risk but have this resistant or resilient signature. And I'll give you maybe an example of a genetic risk that we are aware of. One of the highest risk factors is the ApoE4 gene. We all have copies of ApoE4 or ApoE, and they come in different variations. But people with ApoE4, 2 copies, are at a higher risk, but that doesn't mean that they inevitably develop dementia. And the fact that there are populations of people that do not develop dementia tells us something really powerful, that there's a biological mechanism there that is working in aging that we don't yet fully understand, that's protective. So CureAlz has really concentrated funds in this area and resources by creating what we're calling the Brain Aging Consortium, which is designed to really uncover those protective mechanisms. This consortia constitutes a group of scientists who are interdisciplinary, working together, sharing resources and tools and findings. And they're studying this angle of resilience and brain aging in a healthy way by looking at metabolism, inflammation, vascular health, cellular stress for some. And so the goal for this consortium is to really move beyond looking at Alzheimer's as a single pathway view, but towards a more complete understanding of how the brain ages well. I don't know, can I share another exciting area?
Sure, please do.
All right, so then my next exciting area that I'm really focused on for the upcoming year with the team is really understanding, well, it's like the connection between cancer biology and neuroscience. So cancer has really reshaped the way we've thought about the immune system in general. So over the past decade, cancer has fundamentally focused on immune system and targeted it to understand how you can activate the immune system when it's underperforming, but just as importantly, to rein it in if it's becoming overactive and you get inflammation that's causing cell damage. So some of those tools from cancer that have been developed are CAR T or immune checkpoint modulation. And they show that if you can precisely control the immune system, you can control sometimes the course of disease. So now we're taking these oncology lessons and applying them to brain disorders. And that's really exciting to me because we're learning that actually neurodegeneration really does involve a lot of immune system activation that can either drive forward the disease or potentially even slow down neurodegeneration. And so the way that's happening is through immune balance. And immune balance is really looking at T cells, killer T cells, which go out and attack foreign bacteria, partial fragments of proteins. And then if they're overactive, they can start damaging the remaining healthy tissue, which we don't want. But recently, I don't know if you followed the Nobel Prizes this year, but another type of immune cell was identified and awarded the Nobel Prize, and that was called a regulatory T cell, which is another part of the immune system that restores that balance and sort of calms down the system. So an analogy for how you can think about the immune system in neuroscience might be if your brain was a hockey game, the T cells would be the hockey players, and they're skating hard, they're playing hard in this game, but they're ripping up the ice. The Tregs, or the regulatory T cells, could be the Zambonis that come in, they don't kick out the players, they just clear the ice, cool it down, bring things back into like a stable play, ready to function again, before you destroy the rink. So what we're learning from cancer is that we can manipulate cells to deliberately focus the immune function, normal function. We funded multiple researchers in this area, including David Holtzman, Peter Tessier, Yoni Kipnis, and each of their projects are looking at different aspects of that immune system to either bring in more Zambonis or bring down the immune response itself. So they're really looking at, rather than what if we get rid of the immune system, to a more nuanced question of how do we restore balance? And I think their projects are really important examples of how we can take insights from cancer to really change the new therapeutic directions that Alzheimer's disease research is headed towards.
Thank you for sharing that. Meg, as we both are listening to that answer about the evolution of technologies, I'm curious, with your view as CEO for the last 2.5 years and being on the team for over a decade, how have you seen the role of CureAlz evolve? And are there aspects of the year ahead that you're particularly focusing on?
So, again, I think it gets us back to our ultimate mission. We exist to put an end to Alzheimer's disease, and that doesn't change. But the way we serve that mission, the things that we need to do to advance that mission have evolved, and they continue to evolve. And they evolve in kind of a couple of key ways this year and last year in particular. One is how exciting it is to be in a time where, if you are a early stage Alzheimer's patient, if you have a beta amyloid pathology, some limited amount of cognitive decline, but you do not yet have significant tau pathology, we have a drug, or in fact, a couple of FDA-approved drugs that can alter the progression of your disease. That is extraordinary compared to when I started at CureAlz. And our funding is responsive to that new reality. That demonstrated and validated that beta amyloid is a good target, but it also tells us, because those drugs have had limited benefit, that we need to continue to invest in other targets. And so our funding is responsive to that. We are continuing to look beyond beta amyloid, while we at the same time try to think of better ways to go after beta amyloid. And that's absolutely an evolution in response to the existence of these drugs. But at the same time, what we need to be doing also needs to respond to the circumstances around us. We are thrilled that a bipartisan effort has produced a congressional budget that actually protected the levels of NIH funding for this year. But we are concerned about the way that money is going to be distributed, that we have had an incredible loss of internal career talent and knowledge within NIH and FDA. And that we are hearing from the administration that there is interest in distributing the money to think about geographic distribution as opposed to necessarily the best Alzheimer's science. So our funding will need to be responsive to the changes in federal funding, because all American scientists are fundamentally in need of that federal lifeline. And so we will need to change what we do to make sure that we keep the best and the brightest in the field and active and doing the best possible Alzheimer's science. So again, our focus remains the same, but what we need to do in order to serve that focus will evolve and is constantly responding to new knowledge as well as to new context.
So thank you, Meg. As Natasha, as you're thinking about those great examples you gave and what Meg just shared, that we're going to be responsive, can you tell us about the process by which decisions are made? How does CureAlz think about evaluating potential projects to fund? How do we approach this entire system of receiving ideas from researchers and getting them funding?
That's a great question. The process and how we do this prioritization is really where we have impact, and how we can define that impact. So at CureAlz, our process is designed to be both selective and respectful of scientific effort and time. Writing a full grant proposal for a principal investigator or a researcher is an investment of time and intellectual labor that is time that they're not spending in a lab making the discoveries and running along with the people in that lab to help them learn to become scientists themselves. So it's a commitment that we take seriously and we want to be thoughtful about. So in that, with that in mind, we've created a two-step process. The first step is that researchers would submit a letter of intent, which is a brief review of what their proposal is, small two-pager, which is reviewed by an external research leadership group, which is a group of scientists with very deep but wide expertise in Alzheimer's research landscape. So at this stage, the reviewers are really assessing novelty, impact, relevance to Alzheimer's biology, and also strategic fit within our portfolio so that we don't have overlapping projects to a large degree. This allows us to really identify promising and differentiated ideas really early. And as I said, avoids some redundancy in funding portfolio. Once that LOI is approved, the PIs are then invited to submit a full proposal. This would be a more lengthy description of the project that they are intending to do. That project proposal goes through another rigorous external peer review, which is consistent with best practices for funding research. And the reviewers are selected based on their deep subject matter expertise, where they independently would evaluate the scientific rigor and the potential for impact, as well as the planned feasibility. Can they do what the scope of the project is that they've proposed within the budget and the time they've been allotted? So this two-stage structure helps us maintain focus of our resources where they really matter most, while still being fair and efficient with investigators' time. And we like to prioritize early ideas, bold, unconventional concepts that might be overlooked in a longer partnership sort of process that we have with the NIH, where these traditional mechanisms sort of demand much more preliminary, many years of research before they might be considered for funding. So we're really complementary to that. And at the same time, this process that we have is very rigorous with peer review and ensures that even these high-risk ideas are grounded in really strong experimental design. So the result that we're creating in our portfolio is diversified, one that might have near-term mechanistic insights, but also invite entirely new directions for the field to consider. And so with that balance, I think we're pushing innovation without compromising any rigor in the science itself.
Thank you so much. Well, I appreciate the questions and answers we've already done, but we have a lot from our audience that we received ahead of time and that are coming in through the Q&A. So I will encourage folks to continue to submit your questions through the Q&A. We will get to as many as we're able in the time we have allotted. So let me start, Meg, with a question that I think might help set the table a little bit for some more of this conversation. And that's simply, can you address a little bit about the staging of Alzheimer's diagnosis, how we think about the progression and staging, and make some comments about the current drugs that you mentioned earlier and how the staging of the disease impacts who is able to access those drugs?
Sure. So there's a lot in that question. So I'm going to try to address it somewhat succinctly because I know we do have a lot of questions. But disease staging is really shockingly new in Alzheimer's disease. we didn't really realize for most of the, more than 100 years we've known about this disease, that beta amyloid pathology begins to accumulate decades potentially before there's any emergence of cognitive symptoms whatsoever. And that presents both a challenge and an opportunity. It means that the very first pathology is accumulating in someone's brain before there is significant neurodegenerative damage, before there is significant loss of cells and tissues in the brain that would be essentially long-term damaging for the circuitry that underlies memory, behavior, action, everything our brains can do. So there's a window of opportunity where we hypothesize that if we stopped beta amyloid from continuing to accumulate, it would stop what we call the Alzheimer's cascade. You would not then see other aspects of the pathology even begin to cause damage, nor would you see the damage that then translates into actual clinical symptoms. But that also means that that window can go undetected. There's no reason for us to necessarily be able to look at somebody and say, oh, that person's probably got beta amyloid in their brain, because nothing in their behavior or their cognition will have changed yet. So we really need ways to diagnose people long before there's any behavioral reason to think that we should be diagnosing them, to think that we should be identifying them. And we need to come up with better language. We don't really want to talk about somebody as having Alzheimer's disease if they don't have any cognitive symptoms, because there's so much stigma associated with that. And we don't want to discourage people from getting that diagnosis. So there are lots of terms that get thrown around, prodromal Alzheimer's disease, for example, which are not particularly lay friendly. So we're not hoping to see that kind of language adopted. But what we would like to see is that when people go see their general practitioners for regular healthcare, which we hope people are doing, that they'll actually start getting a brain check along with the rest of their body being checked as a form of preventative care, so that we can start intervening when all that somebody really has is at the beginning of the accumulation of beta amyloid. They don't yet have, for example, the neurofibrillary tau tangles that are kind of the second stool or second leg of the three-part stool. When we talk about Alzheimer's disease, we talk about beta amyloid, we talk about neurofibrillary tau tangles, and we talk about neuroinflammation leading to neurodegeneration. Neuroinflammation is what Natasha was talking about earlier, where it's essentially your body's response to natural processes going awry here. And that's why there is such a similarity to cancer. You can develop cancer in response to infection, but cancer isn't caused by a germ fundamentally, and neither is Alzheimer's. But your body responds to natural processes going awry with an inflammatory response. So to get us back to the staging question that kicked us off, we have this very long period of time where we've got beta amyloid beginning to accumulate in somebody's brain. And then the next stage that we talk about generally is when that beta amyloid actually induces the development of neurofibrillary tau tangles. We don't really have a great handle on exactly what that mechanism is. And we're funding a lot of research into that question with our Alzheimer's Tau Consortium and other projects, because that, if we could break that piece of the causal chain, that would be another opportunity to essentially stop the neurodegeneration that actually translates into clinical Alzheimer's. So that's kind of the second stage that we talk about. And then the next stage is when somebody has both beta amyloid and tau pathology, and we are seeing the loss of material in the brain that we describe as neurodegeneration. And that's going to be a much harder stage to address therapeutically because we have so many different things going on at once. Right now, the drugs that we have approved by the FDA are really only helpful for folks who are in a very early stage where they have beta amyloid accumulation. They may have some early stages of mild cognitive impairment, but they do not yet have significant tau accumulation.
Could you speak to the status of clinical trials that are looking at those people who are not yet symptomatic, but are receiving some of these anti-amyloid treatments?
So those trials are underway and they involve thousands of people. So once they do read out, we are really excited to see what we learn from them. We do expect readout of a number of those in the next couple of years. And the companies that make the current anti-amyloid immunotherapies are, of course, really excited with the hope that their products will actually help people at earlier stages and essentially prevent what we think of as Alzheimer's cognitive change altogether. So we're looking forward to seeing those soon. There are some other companies that are going to be new entrants into the market that are also addressing this space. And there are a couple of other possibilities that are taking different approaches as opposed to the anti-amyloid immunotherapies that the current drugs that are the class that the current drugs are in. So we're excited to learn more. We're going to learn a lot in the next few years. And we are also really excited to see what we're learning in terms of risk reduction strategies with lifestyle choices, as well as environmental exposures. And those absolutely need to be talked about because there is a real hope that if we were able to identify and empower people with what the best choices are, that we could actually significantly reduce the number of people who will eventually need drugs.
Thank you so much. So Natasha and Meg both, but Natasha, I'll start with you. We received several questions that speak to this second thing that Meg was just referencing, and that is lifestyle. Things that folks can do now to reduce their risk. So Natasha, I'll ask you a couple of particular questions, but I'll ask both of you to maybe make some comments around what we're supporting, but also what's happening in the field related to that. So Natasha, we received a question about the Johns Hopkins study around brain games that might be able to reduce the risk of developing dementia. I'm wondering if you could make a comment about that particular study and maybe this broader concept, because we received multiple questions about brain games.
Yeah, I imagine you would, because the titles and stories of the news stories about this have been really captivating, and really suggesting that there's a 25% decrease in dementia or that there's a change in the timeline. And the training, so the study really doesn't actually say that. What it does say is that these training sessions where you're looking at fast processing was associated with a reduced rate of diagnosis over 20 years. However, that study has important limitations because what it does not clearly define is what that means. It doesn't quantify the delay in the years of onset. It doesn't establish what the reduced lifetime risk is. And it doesn't actually confirm the type of cognitive decline that they were looking at because it doesn't look at biomarkers. So what they were really measuring is the time to first diagnosis, which was measured through a hazard ratio. I wish I was a statistician to be able to really explain hazard ratio in a way that makes sense, but it's a measure to look at relative speed of events occurring across the groups. So there was a significant but small change in the groups that they compared, which was a group that was looking at speed processing of this game that causes you to move your eyes a lot, or memory recall, so asking you to make a list and mnemonics to remember how that works, or logic and reasoning group. So those were the three groups. The logic and reasoning group would be looking at ordering patterns and things like that. And so it could be that the reason why you see a significant difference in one whole group, not at an individual level, is that we don't, as we age, normally do speed processing unless you're playing pickleball every week. You're not doing a lot of the sort of tasks that require you to do this sort of swift eye movement changes versus memory tasks we're doing all the time, even as we're aging. And we're doing a lot of reasoning, even as we age. So those group effects are sort of smaller in that memory group and in that logic group compared to the speed processing group. So the normal version, like what is the normal, is hard to really identify in that study. The other significant limitation to consider in that study is the participants and the participants' ability to stay within the program for the entire time. So there is a reduction in the number of people, or attrition in the survivor bias of people that would be continuing in that study for those 20 years. So it could be that the people that may have been diagnosed early passed away earlier than they were able to be diagnosed. So the direct modification of Alzheimer's through brain games is not necessarily demonstrated clearly through this process. I think further studies need to be conducted that would actually be measuring risk assessment and time of onset delay, truly, rather than time of diagnosis, which is also subjective to, if you're healthier, maybe you're not going to the doctor as often and getting checked. So your diagnosis is later. That doesn't necessarily mean it was because of the type of games that you were playing.
Folks have also asked questions about other sort of news headlines. So I'll ask either of you to maybe help us think about, we had questions about lithium, caffeine, supplements, these various ways that people read about in mainstream media to learn about potential options or avenues to reduce their risk. Could you maybe talk about some of those topics that we hear often about?
So I'm going to start with I'm going to start by suggesting that one of the most important things we can all do as news consumers is recognize the limits of headlines. So as Natasha was just describing, the headlines about this brain training study were really exciting. And then if you dig down a little further, you realize that the findings were a lot more limited. And so it's really easy, as we all look at our news feeds, to see a headline and get super excited about it and not have the bandwidth, frankly, to dig deep. What I would say is that right now there is a lot of data accumulating across a lot of studies that do tell us that lifestyle choices make a difference, and that there are broad categories of interventions, as Natasha described to me earlier today, doing what your mom always told you you were supposed to do, that really do affect our Alzheimer's risk. And the bulk of the evidence continues to support that. So sleeping well and regularly, eating unprocessed or less processed healthy foods and doing so on a regular basis, getting good exercise that gets your heart rate up, staying engaged with the world. These are all things that are not that exciting in a headline, but that the bulk of the evidence tells us, and wonderful new evidence with much better studies than used to exist, tell us really do make a difference. The specifics are often less exciting. So to use the example Natasha was just describing, is our brain speed processing games the best way to protect your brain against Alzheimer's and dementia? We really can't say that right now, but we can say that staying intellectually stimulated has shown benefit across a lot of different studies in a lot of different ways. So I really want to both be encouraging and discouraging when people read the headlines about lifestyle. Focus on the big picture and focus on big categories because they do make a difference. Trying to identify the most perfect, most effective, specific intervention is currently beyond what the data tells us. I will jump in a little bit on some of those other headlines, just because I think, in particular, that lithium study is tough because we all know that, or many people know that lithium is already available as a primary treatment for bipolar disorder. And so when we see lithium as a potential Alzheimer's treatment, we get excited because it seems like it'll be available quickly because it already is. The reality is that was a mouse study, and it was a form of lithium that is importantly different from the form that is actually used to treat bipolar disorder. And there are lots of ways that the form that was studied in those mice will be chemically and biologically differently active in a human than the form that is already available in the market. So unfortunately, while there is more study merited to look at that particular form of lithium, there's other data that suggests it's not that exciting as a potential candidate, and we cannot assume that it will be effective or safe just because we have another use for lithium, a different form of lithium that is both effective and safe.
Thank you. Meg, I'll stick with you for a moment on one more of these sort of lifestyle things. We received a question about, have there been studies on certain honeys and types of honey that affect short and long-term memory, hippocampal health, and reducing the load of beta amyloid in the brain?
So I have never seen a large, well run study that would say honey is an appropriate intervention for Alzheimer's disease. That does not mean that honey is not healthy. It does not mean that people shouldn't eat healthy or eat honey. But there are kind of two questions when we talk about honey that need to be separated out in terms of general health before we even get to Alzheimer's. The first is, should I eat honey at all? Well, if you're a person who, for medical reasons, according to your doctor, needs to be careful about their sugar intake, honey is a form of sugar. So it is absolutely going to fall into the category of things you need to watch because your doctor's already told you to watch sugars. On the other hand, if all you are asking is honey a better sugar than other sugars that are more processed? Well, there's a lot we don't understand about sugars and about all of the different micronutrients that exist in whole foods versus in heavily processed foods. But the bulk of the evidence strongly suggests that less processed foods are better for us. And so if you're going to compare sugars, a less processed form of honey is likely to be, therefore, better for you than a highly processed, white granular sugar. So those are two questions before you ever get to Alzheimer's. Then we get to the question of, is this a beneficial intervention for Alzheimer's specifically? Again, I am unfamiliar with any clinical trial that demonstrates honey as an effective intervention in Alzheimer's. I am also unfamiliar with any epidemiological evidence that says that communities that eat more honey, as opposed to eat less honey, different from eating more or less sugar, have less or lower rates of Alzheimer's disease or even dementia in general. So what does all that boil down to? Honey is in many ways a whole food that just as we generally talk about whole foods as being healthier, we would expect honey to be as well. But we cannot at this point say whether taking in large amounts of honey is a therapeutic option for Alzheimer's, either as a prevention or as a treatment.
Thank you so much. Natasha, we received very many questions about blood tests. I'm wondering if you can talk a little bit about what the blood tests that are currently available are actually measuring, and then I'll ask a few questions about how they're being used, what their use is in the clinical trial setting, and what is available to somebody who might want to take this for their own use with their doctor.
I like that you added for their own use with their doctor. That's always the first step. So there are blood tests that are available, and these blood tests are becoming more and more affordable and faster and widespread. The vast majority of these blood tests measure are biological markers associated with Alzheimer's disease, which include normal forms of tau and amyloid proteins. These tests are becoming more increasingly accurate and are being incorporated into clinical settings, especially if you have already memory symptoms. That's where these tests are really useful for understanding differential diagnosis. If you have cognitive decline and you take this test, it can really allow you to have a conversation with your doctor about the type of therapies that might be helpful specific to you and that cognitive decline that you're experiencing. The tests are not predictive or definitive of whether you will or when you may develop symptoms. So they need to be really carefully explored if you don't have any cognitive decline right now. I mean, in my mind, I really equate preclinical use of blood tests at this point to measuring A1C or your cholesterol level. Doesn't mean that you're going to get diabetes or heart disease, but it's a risk factor that you can start really incorporating some of those lifestyle changes that Meg was talking about, and empowering yourself to take control over some of the factors that you can modify, independent of having that sense that you've been diagnosed with Alzheimer's. It's not a diagnosis. It's really a risk assessment.
Thank you so much. I'm wondering if you can tell us about how we know how it's being used right now for clinical trials, and what their relationship is to individuals who might either want to join a clinical trial, or that the clinical trial, its design itself, uses these tests.
These blood-based biomarkers are a tremendous asset to us in understanding how to stage clinical trials. They've made clinical trials sort of a global preventative measure, in a sense, that being able to test early allows you to enroll people earlier with a lower barrier. So previously you would have to do a PET scan, which is costly and timely and kind of geographically limited in terms of access. So blood tests allow greater participation, earlier enrollment, and a more egalitarian enrollment process of people. It also makes trials much faster and more efficient because you don't need to be staging with PET scans throughout. You can take blood tests and pair those with cognitive tests over time. So that makes getting to an answer much quicker, which is what we all want out of our clinical trials. And we're moving towards these blood tests becoming more precise in the future, with the type of selection of individuals that would be brought into one clinical trial versus another. So it's really sort of leading towards precision medicine and stratifying the disease. So it's shifting clinical trials from being symptom-based to really risk-based biology. And I think that these clinical trials are really going to be more accurate and faster for the fact that we have these blood-based biomarkers that we can rely on now.
Thank you so much. So we have several more questions. I'm hoping that we can get to as many as we can in our last sort of minutes that we have together. So I'm going to throw a couple out. None of them are short answers, so we will do our best to get to what we can. But Meg, there's a question in the chat about vaccines. And I know that this itself is a very complex subject. So I'm wondering if you can address some of what CureAlz has funded of the role of vaccines related to Alzheimer's disease, and speak to this as a larger topic of interest with Alzheimer's research.
Yeah, so the vaccine topic is another one that's really exciting because there's such an opportunity here. It started, as it often will in Alzheimer's and other disease research, with epidemiological findings. So when we look at large populations, hundreds of thousands of people across lots of different countries with lots of different vaccine schedules, what epidemiologists started to recognize was that certain countries that had certain vaccines in kind of common access were showing lower rates of dementia diagnosis. And, you could argue, well, that's just better access to healthcare, or it's people who choose to go to the doctor more, or you can imagine all sorts of different factors that could feed into that. And that's one of the challenges with epidemiological research. It's always very noisy. There are always lots of confounding factors. but it tells you scientists where to start asking tougher questions. How do we experimentally start to kick the tires on some of the observations from these studies? And so scientists started to recognize that these associations were popping up unexpectedly, and they started to explore, are these real? And we got involved early on because we had some researchers who came to us and said, one of the vaccines that seems to make a difference is something called BCG, and it is a tuberculosis vaccine. It's not used in the United States because tuberculosis is not endemic here. And so we don't give a vaccine where most people aren't going to get exposed to a disease. But it was recognized that countries that did widely use the BCG vaccine were seeing differences in dementia rates. Interestingly enough, this vaccine is actually a frontline treatment for a form of cancer as well. And there are essentially two typical treatments for this cancer. One is BCG vaccine and the other is something else. And the patient profile that gets one versus the other isn't particularly different. It's a little bit, what does your particular doctor or medical institution do? And so a natural experiment occurred where people who had this form of cancer were treated with one of these, with the BCG vaccine or the other treatment. And what they saw when they looked later, again, this was a retrospective study, was the people who had received the BCG vaccine as this cancer treatment ended up with lower rates of dementia diagnosis later than the people who got the other treatment. And we funded research looking at this data and saying, wait a minute, that is a very unexpected finding. These two groups of patients were essentially equivalent other than the treatment that they got for their cancer. Why would this be? So the first research team that we ever funded in this area was looking at exactly that. What do we see? And what can we learn from the outcomes for these people? And can we start to think about what the mechanism of action might be for this difference? But since that initial work was funded, we've now funded work that looked at other aspects of vaccination because we're seeing the same kind of data, although not as natural, as convenient a natural experiment with other vaccines, including recently a really exciting natural experiment that happened in Wales where people either did or did not get the shingles vaccine based on their date of birth. With people on, the weeks and months of opposite sides of this one bright line birthday having different rates of dementia depending on whether or not they were able to access the shingles vaccine. So we are continuing to fund research trying to understand what the mechanism of action is, because of course we would like to have a therapeutic that is specifically achieving the dementia difference without necessarily exposing people to vaccines for specific diseases. But what we are seeing in the data is that people who get more vaccination events across a whole bunch of different diseases, including the flu and shingles, actually do have lower rates of dementia, and they seem to have different rates of Alzheimer's pathology as well. So this is really exciting because anytime we can tell you about something that's already good for you for another reason that might also help reduce your risk of Alzheimer's is more bang for your buck. And so we really want people to know about this emerging research so that they can make informed decisions every year when they're faced with the opportunity to get a vaccine to understand what all of the possible benefits might be.
Thank you so much. So for those who asked questions that we didn't get to, our team will do our best to answer those separately offline after the event. So in the coming days, you'll hear from us. If we know your name in the Q&A, we will be sure to follow up with you with some of your specific questions. Sorry that we weren't able to get to everything, and thank you everyone for all of your fabulous questions. And thank you, Meg and Natasha, for sharing your thoughts and remarks with us. As we wrap up, I wanted to give you the opportunity to share any final remarks or final thoughts. Natasha, is there anything that you would like to share?
Just that I'm really optimistic for all the science and the outstanding investigators that we're supporting, that are asking smarter questions, better models. And our role at CureAlz is really to be thoughtful stewards of the momentum and to make sure that we're investing in creative science that's going to move us further towards that goal of curing Alzheimer's.
Thank you. Meg, any final thoughts to share?
I want to thank everybody for joining us today and for choosing to be a part of the solution. We will not end Alzheimer's without great science answering really tough questions. And we can't do that without the support and participation and engagement of everybody who joined us today. So I'm just really grateful for that.
Thank you, Meg. Thank you, Natasha. Thank you for your insights, and thank you for sharing everything you did with us today. For our audience, thank you for joining us. We will share the recording of the presentation with all of you as soon as it becomes available. If you have any additional questions or would like to connect with someone at CureAlz, please contact us at rsvp@curealz.org, and we'll be happy to get back to you. We are deeply grateful for your continued support and partnership in the fight to end Alzheimer's. I hope you all have a wonderful day. Thank you.